Dear students, welcome to our official YouTube channel SRN BGNR Government Arts and Science College, Khammam. Myself, Dr. B. Satish Kumar, Assistant Professor in Biochemistry. In last few classes, we have been studying about the molecular biology topics, and this molecular biology paper is in the syllabus of final year students semester 6 of BSc biochemistry students, and of course almost not almost but every life science course syllabus is having this molecular biology paper as one of the major subject, because this will deal with the life process like replication, transcription, translation, regulation of gene expression. So these are very much basics to understand the life sciences at molecular level. Okay, so in last few classes we have been studying the molecular biology lessons, and we have covered the first unit pertaining to replication and transcription, and then post transcriptional modifications. So these three topics we have covered in the first unit, and in the second unit we have covered genetic code and translation, then post translational modifications and inhibitors of protein synthesis. So all these topics we have completed in the second unit, and now in the third unit, in previous class, in part 1 video, that is just previous video lesson, we have studied about the concept of operon, and what is induction and what is repression, and so the lactose operon we have discussed about that, and regulation of the lactose operon. Okay, so far we have covered these basics in the third unit, that is regulation of gene expression. Now let us quickly recap the last video lesson, and then we will start the next topic, that is tryptophan operon. Okay, so in the syllabus we have two subtopics, lactose operon and catabolite repression, tryptophan operon and its attenuation. So these four subtopics we are going to cover in a few video lessons. Okay, so now let us start with the gene expression. So we try to understand what is regulation of gene expression, what is meant by regulation of gene expression, because the gene expression is nothing but expression of gene to its protein product. Okay, so the gene itself contains nucleotide sequence. That nucleotide sequence codes for a polypeptide chain. That nucleotide sequence codes for amino acid sequence in a polypeptide chain. So how the nucleotide sequence from the amino acid sequence is formed from the nucleotide sequence, that is at the gene level. So from the gene to protein level, how the information is passed, that is what the gene expression. So in the gene expression we will study two important processes. First one is transcription and second one is translation. So from genes to protein, what happens in the middle of these two processes, we have mRNA molecule. Okay, so that is why gene expression is nothing but the transcription plus translation. So when we look at the central dogma of life, we have DNA. So from the DNA, what happened? DNA is nothing but here the gene. From the gene, gene X, we are getting mRNA molecule by transcription process. Okay, and after that, this mRNA entering into the translation to form the X protein. Okay, from X gene we are getting X mRNA, and from the X mRNA we are getting X protein. So these two processes are combinedly known as expression of gene. Okay, so now let us see what is known as regulation of gene expression. The genes must be regulated, so that means they must be switched on or switched off based on the need of the organism. Otherwise, if always it is in switched on form, then what happens? The um, cellular energy will be wasted. Like any suppose uh, if we put on each and every light in the house, then what happens? If it even if you even if we are not using, and still if we switch on those lights, that will waste the electricity. In the same way, so based on the need, the cells will switch on some of the genes and switch off the remaining genes. Okay, so when the X is needed, then X gene will be switched on. Then Y is needed, then only Y will be switched on and X will be switched off. So in that way, some genes are switched on and some genes are switched off based on the need of the organism. Okay, so that switching on and switching off mechanisms are known as regulation. So either we can switch on or switch off, and at the same time we can increase the rate of the synthesis and decrease the rate of the synthesis also. So that is called regulation. So uh, the best studied example of regulation of gene expression is the lactose operon. Jacob and Monod, two scientists who investigated the lactose operon, and they proposed the regulation of gene expression theory by taking the example of lactose operon. Okay, so in operon concept, and operon concept is nothing but if you see the lactose operon, the lactose metabolism, uptake and metabolism of lactose requires three enzymes, three proteins. Okay, suppose if we take the bacterial culture of E. coli cell, and if we add lactose to the medium, then what happens? First of all, that take up the lactose into the cell, then it will hydrolyze the bond between the glucose and galactose. The glycoside bond will be hydrolyzed, so that the glucose and galactose will be separated. Okay, so this way it requires two important processes. First one, uptake, and second one, hydrolysis. Okay, for this purpose it requires three different proteins. Okay, so those three different proteins are synthesized by three different genes. They are known as lac Z gene, lac Y gene, and A gene. So lac Z, Y, A. So these genes are known as structural genes because they are participating, the gene products are participating in the metabolism of lactose. Okay, so these are the essential enzymes for the metabolic process. Okay, they transcribe and translate to form the end product, that is the proteins. Okay, so these are known as the three structural genes. So these three structural genes are present side by side in a sequence. Okay, so it became like a cluster of genes. So these three genes are acting as a cluster of genes, because if the that lactose is present in the medium, then these three enzymes or proteins are definitely necessary. So that is why the presence of lactose must switch on all these three genes. So therefore only one switch is there, to switch on all the three genes, and suppose if there is no lactose, then that same single switch is switched off to switch off all the three structural genes. Okay, but in case of eukaryotes, you won't find these kind of operons. Suppose if we take this same example, just for the sake of example, that LAGZ gene will be in one chromosome, and Y gene may be in another chromosome, and A gene may be another different chromosome. They may be scattered around the genome at different places. So for that, what happens? It requires switches for all three different structural genes. No single switch is sufficient to switch on or switch off all the three structural genes. So therefore, in prokaryotes, the prokaryotes always uh, try to maintain the size of the genome at lower level. So it try to have a less amount of genome. It try to decrease the genome size without affecting the functioning of the genes. Okay, so therefore there are no introns in prokaryotes. If you observe that, we have discussed in post transcriptional modifications where the splicing takes place. So the splicing post transcriptional modification takes place only in case of eukaryotes, not in case of prokaryotes, because there are no introns present between the exons coding regions. Exons are nothing but coding regions, so there are no non-coding regions and no repetitive structures, all those things in eukaryotes. So that is why it is try to maintain less amount of genome size. Okay, so that is the one of the characters of prokaryotic cells, because their lifetime is very small. Within 20 minutes they get double, and so for that, within very short life, they have to synthesize all the molecules uh, to form two daughter cells from a parent cell. Okay, so so many number of metabolic processes takes place simultaneously within the span of 20 minutes. So that is why it always try to keep its size as low as possible. Okay, so therefore we will see all these uh, genes which are useful, which are useful to carry out a specific function like a metabolic process. they are they will form like a cluster, and that cluster is under the regulation of some regulatory elements. So now, along with the regulatory elements and the cluster of structural genes is known as the operon. So in case of lactose, we have structural genes, three structural genes, and the regulatory sequences. So regulatory sequence, one gene is also there, that is LAC-I gene, that codes um, a protein called repressor protein, and we have promoter region and operator region. So operator is the place where the switch is located. Okay, operator itself is the switch there. So repressor, in the absence of lactose, what happens in the absence of lactose? The I gene expressed to form the repressor, which is active repressor. Now that active repressor binds to the operator region. So operator region is in fact located within the promoter sequence. Okay, so now here I have shown it like the operator is in front of the promoter. So that is why that RNA polymerase is unable to bind to the promoter. Even if it binds to the promoter, it cannot move into the structural genes and transcribe them, because this is stopping the movement. This is restricting the repressor molecule, restricting the movement of RNA polymerase. So now it is in front of the polymerase enzyme. Okay, So that is why it will inhibit the enzyme, that is RNA polymerase, from transcribing the structural genes. Okay, so once we add a small amount of lactose to the medium, then what happens? Even though there is no lactose earlier, even though there is no lactose in the medium, the lactose operon can produce very small quantities of enzymes. Okay, very less, low level of expression takes place, even in the absence of lactose also. That is almost negligible amount of uh, mm, these structural gene products will be formed. Okay, because of that we will get a small amount of beta galactosidase enzyme and permease that allows the entry of lactose into the cell, and transacetylase, which will detoxify the materials, unwanted materials which entered into the cell along with the lactose. Okay, so now at when the moment we add the lactose to the medium, then what happens? The cell is having very less number of permease molecules, so therefore it allows a small amount of lactose to enter into the cell. Okay, so now small amount of lactose enter the cell because of less quantity of beta galactosidase. That beta galactosidase converts lactose into allolactose. Okay, this is the allolactose. So in fact it should break this glycosidic bond, and it must form that galactose and glucose from the lactose. But because of small quantity of beta galactosidase, it converts the lactose to the allolactose. So the difference between lactose and allolactose is just in lactose we can have that 1,4 glycosidic bond. Okay, so this is 1 and this is fourth carbon. Okay, 1,4 beta 1,4 glycosidic bond in lactose. But in case of allolactose, we have 1 and 6 1 6 glycosidic bond. Just the bond has been shifted from the fourth position to sixth position. So because of that, 1,4 glycosidic bond becomes 1 6 glycosidic bond. Okay, there is nothing difference more than that between lactose and allolactose. So allolactose is formed. So now this allolactose, okay, so this acts as inducer. Okay, this acts as inducer, which inducer means that will induce the structural genes, that will switch on the structural genes. How they will switch on the structural genes? They will that inducer will inactivate the repressor. Okay, here the inducer binds the repressor so that the repressor becomes inactive. So now the inactive repressor cannot bind to the operator site. So once the operator site is free, then the promoter site will be occupied by RNA polymerase. Now the RNA polymerase has no obstruction in front of it, so that it moves along the structural genes and transcribe the structural genes to produce a large mRNA molecules. So we can observe here that large mRNA molecule is a polycistronic mRNA. So that means in this mRNA we have Z region, Z portion and Y portion and A portion. In fact, in eukaryotes, each and every gene produces one mRNA molecule, one corresponding mRNA molecule. But in case of prokaryotes, because these prokaryotic genes are in the form of a cluster, and the structural genes are in the form of cluster, they all transcribed at a time, and it produced a large mRNA molecule which is polycistronic mRNA molecule. Polycistronic means, poly means many, cistron means genes. So more than one genes are transcribed at a time, transcribed as a unit, and it forms a large mRNA molecule. That mRNA molecule is called a polycistronic mRNA. Okay, now the polycistronic mRNA enters the translation process and produce three different proteins. Okay, three different proteins, because we have three different genes in this polycistronic mRNA. Okay, so we have Z region, Y region, and A region. So the Z region is separately translated by one machinery, translation machinery, that contains ribosomes, tRNA molecules, initiation, elongation, and termination molecules. Okay, so now we will get one that Z portion will produce beta galactosidase, and Y portion will produce permease, and A portion will produce transacetylase. So that means I will explain it some more clearly so that you can understand what is the real process that takes place during the translation of polycistronic mRNAs. Consider this a polycistronic mRNA. We have Z region, Y region, and A region. Z, Y, and A region is there. So that means here the initiation codon will be there and the termination codon will be there. Okay, so that is why the ribosome binds here and translate this Z region separately. You will get the Z product, that is beta galactosidase. And for Y region, within the same polycistronic mRNA, we will have another initiation codon and termination codon. So because of this, another set of ribosomes translate this region and produces the Y product, that is permease. And in the same way, the A is also contains initiation and termination codons, and it separately undergoes translation, and we will get transacetylase protein. Okay, so that is what the translation process that occurs in case of polycistronic mRNA. Okay, so now that polycistronic mRNA is formed, and from the polycistronic mRNA we will get these corresponding protein molecules and the beta galactosidase. In fact, the switching on mechanism will enrich the quantity of these three proteins. So beta galactosidase concentration increases, permease production also increases, and transacetylase production also increases. So because of this, what happens? The permease goes and binds, inserted into the cell membrane, and that allows entry of large quantity of lactose into the cell. Okay, more permease molecules, then more amount of lactose enters the cell. And because of more quantity of beta galactosidase within the cell, that hydrolyze the lactose into glucose and galactose. So that glucose and galactose further enters other metabolic pathways where they are oxidized to form oxidized to produce energy and other materials necessary for other metabolic processes. Okay, so then the transacetylase, so this is a detoxifying enzyme which detoxify unwanted materials which entered through the permeases along with the lactose. Okay, so this is what the lactose operon, regulation of lactose operon. So in the presence of lactose, the genes are switched on, and in the absence of lactose, all the genes or operon is switched off by means of, so this is mediated, the switching on switching off mechanism is mediated by one protein product, that is repressor protein. Okay, so repressor is the key protein which regulates the operon. Okay, active repressor, that we should remember. This important point is, in lactose we will have active repressor. Okay, so now then so that is about the induction process, and second process is repression. Repression is nothing but switching off. Okay, so if we switch off the genes which are already in switched on status, okay, so that uh, process is known as repression. So repression means some genes are already in switched on state, and some signals leads to switching off of the switched on genes. So this phenomenon is called repression. Okay, so the best example to explain the repression process in prokaryotes is the tryptophan operon. So before going to learn about the tryptophan operon, let us consider, let us see what is tryptophan, and what are the structural genes, what are the function of structural genes of tryptophan operon. Okay, so as we all know that tryptophan is one of the essential amino acid. Okay, in fact these essential amino acids are not synthesized by higher organisms like human beings, but prokaryotes can synthesize all the amino acids, so all the 20 different amino acids. Okay, so here tryptophan, the aromatic amino acid, which has a little bit complex structure. So that is why it during the biosynthetic pathway of tryptophan, it requires several different enzymes. Okay, so in a metabolic process requiring several different enzymes, the tryptophan will be synthesized. Okay, so here if you take an E. coli cell, if you take an E. coli cell, that E. coli cell continuously synthesizes several hundreds, even thousands of polypeptide chains for its for all the life processes. So that is why all the all the 20 different amino acids must be synthesized based on the need. Okay, so that means the E. coli cell requires all the 20 different amino acids continuously. Continuous supply of 20 different amino acids are required. Okay, so that is why the E. coli cell continuously synthesize these 20 different amino acids, okay, which are part of all the polypeptide chains. So, so here we are going to study only one of those 20 different amino acids, that is tryptophan. So tryptophan is synthesized by five different, in fact three different enzymes. Okay, in fact we require more than three enzymes, but uh, the precursor molecule, the precursor molecule of tryptophan is chorismate. Okay, from the chorismate tryptophan will be synthesized. Okay, so therefore from chorismate to tryptophan we require five enzyme activities, and those five enzyme activities are shown by three different enzymes. Okay, three different enzymes, and they will have five different enzyme activities. Because of five different enzyme activities, the chorismate is converted to tryptophan. So if you look at the biosynthetic pathway of uh, tryptophan from chorismate. Okay, so now chorismate is here. From the chorismate we will get anthranilate, and from the anthranilate we will get PRPP anthranilate, and from that we will get another in carboxyphenyl amino deoxyribulose 5-phosphate. So all these things takes place, and finally we will get the tryptophan. Okay, so here we have 1, 2, 3, 4 and 5 enzyme activities are required to convert chorismate to tryptophan. Okay, so these five, in fact these five enzyme activities are there, but this one and this one is uh, these two activities are part of a single enzyme. Enzyme 1, say for example enzyme 1 is responsible for these two enzyme activities, and enzyme 2 is responsible for activity of these two enzyme activities, and this is enzyme 3. So we require enzyme 1, enzyme 2 and enzyme 3, three enzymes. Okay, but this enzyme 1 is having two subunits, one subunit for this activity, another subunit for this activity. Enzyme 2 is having this subunit activity and this subunit activity. So uh, completely we require 5 subunits. So from the 5 subunit we will get 3 enzymes, and these 3 enzymes are essential for the conversion of chorismate to tryptophan. So that we are going to see now. Okay, so this is about the tryptophan biosynthesis from chorismate. So let us look at the structures here, so you can easily understand. So we have chorismate is here, we have chorismate here, and from the chorismate we are getting anthranilate. So enzyme 1, enzyme 2, enzyme activity 2, and then enzyme activity 3 and enzyme activity 4, and so after that the enzyme activity 5. Okay, so from these 5 steps we are getting tryptophan from chorismate. Okay, so that is why we require 5 genes to produce 5 polypeptide chains, and 5 from the 5 polypeptide chains we will get 3 enzymes. Okay, so that is the overview of the structural genes necessary for the metabolism or biosynthesis of tryptophan from chorismate. Okay, so here let's get back to this slide again. So you can see that tryptophan is one of the 20 different amino acids essential for the synthesis of all kind of polypeptide chains. So that is why continuous need is there, continuous supply of tryptophan is essential by the uh, E. coli cell. So therefore it is always in switched on form. The tryptophan operon will be in switched on form, always it will be in switched on form. Okay, as long as the tryptophan demand is there, the tryptophan synthesis takes place. Once the production of tryptophan that exceeds certain range, and the availability of tryptophan goes beyond a specific limit, then what happens, that more quantity of the abundant quantity of tryptophan itself acts as repressor and repress that tryptophan operon. Okay, so it is if we observe, if we study lactose operon and the tryptophan operon side by side, you can understand what are the differences between induction and repression. In induction, okay, so now quickly explain and you try to understand the differences between induction and repression. So suppose we have induction at my left, induction, and here the repression. So this is tryptophan operon, this is lactose operon. So in lactose operon, first point is the lactose operon, always it will be in switched off. This is off form, but it is on form because the demand for tryptophan is more. But here it is not synthesizing. The lactose is not being synthesized, but in fact it is being utilized. It undergoing catabolic process. It is undergoing breakdown process. Lactose undergoing breakdown process, but tryptophan taking place in biosynthetic process. Two are completely opposite, so that is why the mechanism of regulation is also completely opposite to each other. Okay, so the genes operon is off form and here the operon is on. Okay, so now we have a repressor that is active repressor to stop the synthesis of to stop the utilization of lactose once the lactose is depleted in the medium. Okay, repressor is active. So in case of this tryptophan, what we can expect? The repressor is there, but the repressor is inactive. So by the time it is synthesized, it will be in inactive status in tryptophan, but in case of lactose it is in active status. Okay, so now here lactose acts as inducer that inactivate the repressor. Here tryptophan acts as repressor that activate the repressor. Okay, that activate the repressor protein. Okay, these are the three major differences and between the induction and repression mechanism. So once you understand the induction mechanism clearly, then it is very much easier to understand and even remember the repression process. Okay, exactly opposite to one another. So now let us see the structure of tryptophan operon. So just like the lactose operon, in tryptophan operon also we have the structural genes, but in this case, as we have already discussed, that it requires 5 different genes to produce 5 polypeptide proteins, and that 5 polypeptide proteins finally form 3 enzymes. Okay, so here we have 5 structural genes. From right to left, if we study, if we see one by one, we will get tryptophan A, B, C, D, E. Okay, 5 genes. Okay, from left to right, we will get E, D, C, B, A. Okay, simple 5 enzymes. Sorry, 5 genes. Okay, so these 5 genes are regulated by the regulatory sequence. So here we have the tryptophan R. R stands for repression. In case of lactose, we have gene I. Here tryptophan R, repressor. So this repressor protein, okay, so that repressor protein will be continuously synthesized and it produces a repressor protein. Okay, the gene produces repressor protein. That repressor protein is inactive. So one by the time it is produced, it is synthesized, it is in inactive form. Okay, so that repressor cannot bind to operator site because that is inactive. Okay, so now what happens? The operator site is free and promoter site binds RNA polymerase and transcribe all the structural genes, and we will get this polycistronic mRNA, and from the polycistronic mRNA we will get 5 polypeptide chains, and these 5 polypeptide chains will assemble into 3 enzymes, and these 3 enzymes will convert chorismate to tryptophan. So that is what happens. Okay, so that is why we have 5 structural genes and one regulatory gene, and like that of lactose we have promoter and operator site. Only difference is the repressor protein that initially synthesized is inactive in status. Okay, so now let us look at the regulation of this tryptophan operon. So what happens in the absence of tryptophan, and what happens if the tryptophan levels are very high? Okay, so in this case the tryptophan levels are low. For this case, we will take this case first. So here the tryptophan levels are low. So that means the cell requires tryptophan production. Okay, tryptophan must be produced to enrich the pool of tryptophan amino acid. Okay, so for that, that structural genes must be active. Okay, so here in this case, what happens? Let us see that tryptophan regulate the repressor gene is active. So we will get that mRNA, corresponding mRNA. From the mRNA, a tryptophan repressor is synthesized. That tryptophan repressor is inactive in state. So that is why it cannot bind to operator site. It is unable to bind to the operator site. Then what happens? Because of that, operator is free, promoter is occupied by RNA polymerase that transcribe the structural genes, and we will get a polycistronic mRNA. From the polycistronic mRNA, what happens? We will get 5 products. So this is the first enzyme component 1, and this is the first enzyme component 2. So now these two polypeptide chains combinedly form the enzyme 1. Okay, enzyme 1. And next, then uh, that next gene, the C will form directly this third enzyme, in fact. Okay, and the second enzyme, that second enzyme, and B and A, these two genes will produce two subunits, alpha subunit and beta subunit of tryptophan synthase, and these two becomes uh, f- uh, bind to each other and form a multi-subunit complex uh, enzyme, multi-subunit enzyme, that is tryptophan synthase. So we have three enzyme activity from 5 different polypeptide chains. First one is anthranilate synthase, which contains two component ones and two components two. Okay. So and second one is indole 3 phosphate, indole 3 glycerol phosphate synthase, and third one is tryptophan synthase, which contains alpha 2 and beta 2 subunit composition. Okay. So now these three enzymes participate in the biosynthesis of tryptophan, which which will lead to the formation of tryptophan from chorismate, like we have seen in the earlier um, slides, that the chorismate becomes tryptophan in the tryptophan biosynthetic process pathway. Okay, so this is what the uh, regulation or that takes place at low level of tryptophan. So now what happens? Tryptophan levels goes up because of the structural gene products. Okay, so if the levels of tryptophan is high, the level of tryptophan is high, then what happens? That high level of tryptophan must shut down or switch off the structural genes. Otherwise, it will continue to synthesize the tryptophan beyond the requirement. So that means it reached a saturation point where the tryptophan, the further production of tryptophan is no longer required. Okay, it should stop the synthesis of tryptophan temporarily. Once it stops temporarily, then what happens? The cell will utilize the tryptophan. Once again, the tryptophan levels goes down, and once the levels are less, so then again the tryptophan operon is switched on. So uh, therefore, the switching on and switching off takes place mm, alternatively. That will ensure the steady supply of tryptophan for the cellular needs. Okay, so this way, like thermostat, if you take the example of thermostat like iron box, what happens, we will adjust the temperature by one regulatory knob. Then what happens, when it is switched on, the temperature of that iron box goes up, and once it reaches a point, then the it will be switched off. Okay, automatically it gets switched off, and once it is switched off, the temperature gradually comes down. Once it reaches uh, lower limit, then again it will be switched on. So this switching on and switching off mechanisms will ensure mm, the maintenance of temperature in a particular range. So in the same way, the levels of tryptophan will also be maintained in a particular range. Okay, above the level, the tryptophan operon is switched off, and below the level, the tryptophan operon is switched on. Okay, so that is what we are learning in this video lesson. And at high levels, what happens? Yeah, we will get the repressor protein from repressor gene, and because of high levels of tryptophan, the tryptophan binds to the repressor and make the repressor active. So now that active repressor binds to the operator site. Here, the tryptophan itself acts as a co-repressor. Okay, repressor and co-repressor complex. It is known as repressor and co-repressor complex. This complex is active. Now this active repressor co-repressor complex binds to the operator site, and it prevents physically the entry of RNA polymerase into the promoter's region. So now the RNA polymerase is unable to bind to promoter site, and structural genes are not transcribed because of uh, that the binding of repressor co-repressor complex at the operator site. So switch genes or structural genes are switched off. So no gene products, proteins, and ultimately the enzymes will be produced, and because of that, the synthesis of tryptophan from chorismate will be stopped. Okay, so therefore at high levels, the genes are switched off. At lower levels, the genes are switched on. So this mechanism is known as the repression. Why we are calling it as repression? Because the genes, the operon, are already in switched on status, and the abundant tryptophan levels just switching off the genes, structural genes. So that is why they are repressing the operon, which is already in active state. Okay, so that is what the repression, and hope you understand the differences between lactose operon and tryptophan operon. And in the next class, we are going to study about the catabolite repression process of lactose operon. Thank you all.